So next is the ingredients. So what a typical reaction of PCR contains the following ingredients. There is the water required, which is 15 microliter, and buffer is required 2 microliter. Magnesium chloride is required 2.5 microliter. DNTPs is required in 2 microliter, and forward primer quantity is required 0.5 microliter, and reverse primer is also required 0.5 microliter, and template DNA in which our desired segment, which could be we need to amplify, is required 2 microliter, and last one is the TAC polymerase, which is required 0.5 microliter. If we add all of these quantities, it will become the 25 microliter, the total volume. The total volume which is present in the PCR tube in which we perform the PCR. So the procedure is done in a PCR reaction tube, which is specially designed to bear the heating and cooling parameters, and so PCR tubes are designed for even heated distribution. Next, if we little bit see if where we extract the DNA for our own interest. So the DNA could be extracted from any body type. We know that every body cell contain the DNA except red blood cells. So from blood, if we extract the DNA, so we can obtain the DNA from the white blood cells. Next is the we can obtain the skin. Skin is also contains the epithelial tissues in which we can obtain the DNA. Next is the saliva. Saliva also can buccal cavity cells also contain the DNA in which we can obtain the DNA. Next is the hair follicles. Also hair follicles also have the DNA, and these are the four main samples from which we can extract our DNA. Next, what are the steps required for the PCR to take place? First, we extracted DNA and pour it into the PCR tube. Next, first primer we insert or pour into the PCR tube, and next step is the second primer, which is also pour into the PCR tube. Next step is the nucleotides or DNTPs also inserted in the PCR tube, and last TAC polymerase. TAC polymerase we added at the last step of the PCR reaction. When all these ingredients are added into the placed into the machine, this machine is called THERMOCYCLER. So THERMOCYCLER, we pointer, where is my pointer pen? Thermocycler. This is the machine in which we can place the PCR tube. This is the plate, well plate, in which we can insert the our PCR tubes. This machine can precisely heat and 54 degree centigrade, then it is 54 according to our required primers, our design primer. When the temperature reach to 54 degree centigrade, then naturally the primer which are complementary with to the our desired DNA segment, and they can bond with their complementary DNA. One is the forward primer which is attached, and you can see that there is the 5' and 3' and which is attached complementary with each other. Go down. Third step is called extension. When the temperature of the PCR tubes in thermocycler also goes again rise, and it will reach into the 72 degrees centigrade, 72, 73, 71 degrees centigrade, according to the certain criteria. And at this temperature, from where the tag polymerase attach with the primers, and nucleotides are also present in the solution. So tag polymerase pick up the specific nucleotide according to the base pair, and extend this segment or this DNA segment into the 5 prime to 3 prime direction, and also the other strand also goes into the 5 prime to 3 prime direction, and DNA polymerase goes on by adding the nucleotides from the solution. And after the reaction, we can say that one DNA molecule divided into the 2 DNA molecule. In this animation, you can see that all of these steps are showing that the first denaturation temperature 94 degree, DNA strand separated, annealing 54, primers are binding, extension, and DNA polymerase extend the DNA segment by binding specific nucleotide from the solution. Point E point. This portion is called nucleus, the cell nucleus. It is called our DNA of eukaryotic gene, and these are the segments of the DNA: exons, introns, exons you mean this is the region of DNA which carrying the gene expression. Next is the real-time PCR or quantitative PCR, qPCR. This is the also new application in PCR technology. So if you see this diagram, this is the real-time PCR. Okay, next is the advantages of PCR: quick, reliable, sensitive, relatively easy, specific. Means if from the discovery of the PCR or invention of the PCR reaction, we can see that if we make the we want to make the many copies of the our targeted DNA, then we clone it into the E. coli, and then it will multiply, then we'll extract this DNA. But after the invention of the PCR technique, it is very quick, reliable, and sensitive also. Means we should take care that no contamination could be occur in this step. So easy and also specific. If we say that what are the disadvantages of the PCR: need for equipment, means we need a thermocycler, which is expensive machine, and we laboratory, and tag polymerase, primers. Tag polymerase is expensive. Contamination, there is a danger of contamination, or the very high chances of contamination. Then no DNA or amplification could occur, and there are dimers or any sort of other contamination are obtained during PCR. False reactions, cross reactions, unspecific amplification. Last is the PCR application from where the PCR could be applied. Human genetic testing and disease diagnosis. If we any test the genetic disease or any disease diagnosis, where PCR could be applied. For DNA cloning, if we want to clone a specific segment of the DNA, then PCR could be also applicable. Also the gene expression in the form of RT-PCR, quantitative PCR, both techniques can be used for the expression of the gene study. Next is the diagnostic applications, and if you like, and if you obtained any sort of knowledge, and you are learn any word from this presentation, so like my channels and subscribe. Thank you very much.